So we're going to start our notes basically by talking about how to put graphs together, so visual representations of the data that you have. So last night you looked at the categorical versus quantitative data. Categorical is what we're going to start with, and categorical is anything you put obviously into a category. So hair color, eye color, political affiliation, anything that is not really numerical. So categorical data, you pretty much always in this class are going to do a bar graph. You can do a pie chart, but we don't really use those. So to make a bar graph, you're going to do a frequency chart, which is literally just counting how many times people show up in each category. So if there's 5 people with brown hair, you know you have a 5 representing the height of that bar. Oftentimes we will have to change that to relative frequency, which is just changing those counts into percentages of the whole, and we'll talk about why here in a little bit. So that's kind of the basic, counting and just literally making bars that height. So let's talk about how to actually make the bar graph. So the one most important thing that you do is that when you make the bars, all of the bars are the same width. So that's the area principle. You want only the height to change the representation of how many things are in that category. So every bar should have the same width. And if you're using like pictures or little symbols to represent something, all those little symbols need to be the exact same size. So it's only that height that is the difference. The second most important thing on any graph that we do, not just bar charts, is that you are labeling. You need to label the x-axis, the y-axis, and anything else like the numerical stuff and anything if you have different categories, so that the person who's looking at it can tell everything they would need to know from the graph. Assume they know nothing when they're looking at the graph. The horizontal axis, that should have the variable name, so if it's hair color, that should be said there, and then the category is labeled as well. You want to make sure you have bars that have space between them, so that means that you can rearrange them. There's no specific order that you need to put the categories in, and that'll be different from the histograms that we do next on the numerical data. And then the last thing you want to pay attention to in making it is that you always start your y-axis, your vertical axis, at 0. And then you can either do your relative frequency as percentages, or you can do counts if you just have one variable going on. Pie charts, a little bit of information here. We're not really going to do that. Just know that if you're doing a pie chart, you cannot have categories that overlap. We're going to look here at a couple different graphs. I want you just to take a second and look to see if you can figure out anything that's wrong with these graphs. So hopefully you paused it. And as you see in this DirecTV graph, the area principle is definitely not used. So that DirecTV is a giant box widthwise, and it is definitely not representative. It's not even like about double the orange box with the cable down here. And so it looks like it's a lot bigger, but it's not relatively, according to the visual representation, it's not accurate. So that graph is misleading for a bar graph. Over here on this one, we have what looks like an increasing some type of trend. However, these are just countries, and so showing the countries as they have the share of GDP is kind of stacking them so you can see the country, but that's not something that should be done in a scatter plot like that. So you cannot use categorical variables in this little scatter plot, which we'll learn about in Chapter 3. And then down here we have some pie charts, and it's like trying to show that there's some increasing happening, but not really sure what it is because I don't know what's increasing. So by the year it looks like it's going up, but that's kind of misleading as to what's going on. So make sure that your graph is simple and to the point and doesn't try to mislead people. So here we have some examples of different bar graphs. So this would be dominant hand between males and females as we're going to compare. So the first one right here, that's just your basic bar chart, and so that's just all everyone together. So you have the number of left-handed people, the number of right-handed people, and then ambidextrous. And you can do frequency there because you just got everybody together, so I know that's 5, and that's 11, and that's 1. Now when we do a comparative bar chart, which is over here, I'm going to split it into males and females, and I want to be able to compare them. Now if I have the same number of males and females, I can go ahead and I can do the frequency. However, if I have like 30 males and only 18 females, if I do counts, that's very misleading to be comparing the two different groups. So in that case, we would need to do relative frequencies. So if you have different sample sizes, you will need to do relative frequency, which I don't have an example of right here. So you can either do them where you do side by side and you need some type of key here to indicate what's what, but you see all the good labeling. You can also do a segmented bar chart, so it's stacked, and you can do the males and then the females or vice versa, so you can kind of compare all of them together. You can also do them sideways, so we could flip them so that the X and the Y axes were different, which is fine. Just label them the same way. And comparative bar charts can have more than 2 variables, so I could have done like age and done like a group of children, adolescents, adults, something like that. So you can have more than 2 groups. And I could have flipped the variables if I wanted to. I could have had a male-female group and done left, right, and ambidextrous. So now we're going to actually look at some data and make a comparative bar chart here. So oftentimes you'll get your data in a two-way table, which is shown down here. So they give you the counts of people who had a full response to St. John's wort and full response to Zoloft and so on. So we're looking for some type of relationship here between the response and the treatment that they were given. So we're looking for an association. So if there was absolutely no association, so if the drug and the reaction was not related, all would have a similar response. So all 3 categories would have similar counts, I guess percentages. 3 categories, similar percentages for each. So each little bar, if you're comparing them, will be pretty similar. Now, it's not going to be perfect because it's just a sample, but it should be relatively close. So we're going to look at, this is a study to see if we had what the treatments for severe cases of depression, moderately severe cases of depression. So they gave people different types of treatments, and I don't know, so if they're doing a placebo, they wouldn't have known which one they were getting. And then they did the counts on how they responded here. So some things to know about the table. So the marginal distributions is talking about the margins. So if I just did like 113 out of 338, 109 out of 338, and 116 out of 338, I get the percentages of the people who took each drug. And that would be considered a marginal distribution. You could also do these numbers out of the 338, and that would also be a marginal distribution just of the responses. So each cell is giving you the count of the combined treatment and response, but if you're doing marginal, it's just talking about one of the variables. So let's look at different ways to ask this question. So blank is the proportion of people who took the placebo and had no response. So they're taking the placebo, they had no response, so that's our 66 right here. So we have 66 out of all the people. The proportion of placebo users that had no response, so now we're only talking about placebo users, so that's only 116, and there were 66 of those 116 that had no response. And then the proportion of people with no response that took a placebo, so we're talking about the no response people, so that's only 192 of them, and they took the placebo, so that's 66 of them. So you can see just slightly different ways to ask the question, very different percentages because your denominator will change. So now we're going to look at what's called that conditional distribution, where we are actually comparing to see if one is better than the other. So did the response depend on which drug they used? So I want you to take a second and pause this and see if you can actually come up with these on your own. I'm going to do the first one and then go ahead and pause it and try to do the rest, and I'll have them filled in for you. So we're looking at just the St. John's wort people, and there were 27 of those with this full response. So we've got 27, and we're only looking at the St. John's wort, so that's 113. So that's 24%. So take a second and try to do the rest of those on your own before you go. All right, so check your answers and make sure you have the same percentages that I do. So remember, you're only doing the totals for each column because you're only looking at the specific group that had that drug. So now we're going to actually make a segmented bar chart to look at this conditional distribution. So make your y-axis into percentages, and we have to do percentages because there was a different sample size of each drug. Okay, so we're going to go all the way from 0 up to, we just need to go to the highest. You can go up to 100 if you want. I'm going to go up to about 55 or 60%, and then we're going to do some labeling on the bottom. So you try to make it yourself, pause it, and check and see if yours matches mine here in a second. All right, go ahead and check yours. So that should be about what it looks like. Now you're doing it by hand, so it's not going to be perfect, but it should generally have all the bars the same width. Make sure you're trying to space your numbers similarly over here on your y-axis. Make sure you did percentages because you have different sample sizes, and so everything is labeled. So just looking at the graph, does it look like the results are the same for all 3 medications? So remember, if they are the same, we should pretty much see same percentages, similar percentages in response for each drug. Now, you do have kind of that the no response is the highest in all of them, and then your full response, and then partial, but the difference in percentages is quite a bit different. So it's seeming like maybe there is some type of difference. Placebo actually had the highest full response, so maybe the other 2 drugs really aren't doing things. So those are things that we'll explore later when we actually run the test, but we can just kind of compare and explain, like literally just telling someone what it looks like. So looking at that graph, I'm not sure I'd be totally convinced that I would recommend someone take anything specifically. Jim, no on it there. So those would be maybe some more treatment or more studies need to be done. So what would the graph look like if the response, if the treatment did not make any difference? So they would all look the same, or at least similar. Again, it's not going to be perfect because it's just a sample. So if it did make a difference, if there was an association, so very different percentages of responses. So in each treatment. So that's what you're going to be looking for as you do some analysis on this stuff. All right, just the last thing we're going to mention, this is not technically something that's going to be tested on the AP, but it's something interesting to know about. So this is Simpson's paradox. So an association between 2 categorical variables can be reversed when you consider an extra variable. So we're looking here at 2 players' batting averages on a baseball team. So 320 versus 298 is their overall batting averages if you do it just against right-handed people, pitchers. B actually increases and is higher. And also versus left-handed pitchers, B is also higher. So A is better overall, but if you split it into right and left-handed people, B actually does better in both situations. So this is a case where the association switches when I add that pitcher-handedness in there. So it's because the player B actually faced a lot more left-handed pitchers, and they are more difficult usually for if he's a right-hander to hit off of. So he ended up having a harder situation to hit in a lot of the time, but he actually hit better in both of those situations, but it brought his overall batting average down because he had the harder situation more often. So it doesn't happen very often. Simpson's paradox is fairly rare, but it's just an interesting thing to take into account.